Today we're going to take a look at acid-base neutralization, review what it is, and then we're going to do some calculations involving acids and bases, but tying them in with reactions, so that means more stoichiometry. And we're also going to introduce an experimental technique called a titration, where you're actually doing acid-base reactions in the lab, and you use your data to do the same types of acid-base neutralization calculations. So first, let's do a review of what acid-base neutralization is. This was first introduced to you probably in grade 10, and then we did a little bit on it back in our second unit on chemical reactions. It's a double displacement reaction where an acid and a base are your reactants. And when they react together, they always produce two distinctive things. One is a salt, and the other one is water. But in chemistry, when we hear the word salt, we're not just thinking table salt, although in this example it is showing sodium chloride. This is going to mean an ionic salt or an ionic compound. So it can mean many different things, and different salts have different properties. When you're dealing with a strong acid and a strong base, so think back to your list of strong acids and bases, any one of those, if two of those react together in the right proportions according to stoichiometry, so what that means is if they react in equal amounts of moles that are required, you end up with a neutral solution or a pH of 7. So in the example that's shown up here, we have a strong acid, hydrochloric acid. We have a strong base, sodium hydroxide. If they both get completely used up, they'll produce salt and water. So at the neutralization point, you only have salt and water mixing. And as long as you start with a strong acid or strong base, this salt will be neutral. Now, the word neutralization, though, is sometimes deceiving because we're not actually talking about neutralized solution after this happens. It's more that we've neutralized all of the acid and all of the base that's there. If you investigate more chemistry past grade 11, you'll learn more about what happens when you have different salts that are produced or when there's weak acids or bases involved. Then the pH of that solution can vary. It can be acidic or basic, but that's going to be for next year. So let's take a look at some problem solving with neutralization reactions. So in this problem, you've got 13.84 milliliters of sulfuric acid that just neutralizes 25 milliliters of a 0.1 mole per liter solution of sodium hydroxide. What is the concentration of sulfuric acid that's involved? So whenever you hear a reaction taking place, you need to have a balanced chemical equation. So again, this is one place where you're going to see your acid nomenclature come back. So you need to be comfortable with that. So let's write the balanced equation first. Sulfuric acid is H2SO4. It's going to react with sodium hydroxide, NaOH. You're going to get water between hydrogen and the hydroxide. And water's a liquid. And then you get your salt. So the salt is from the metal ion in the base, sodium, and the sulfate ion in the acid. So it'll be sodium sulfate. And then you need to make sure you balance the equation. So if you're taking a look, you probably notice that it's not balanced the way that it is. So to make sure you balance it, you need a 2 in front of sodium hydroxide and a 2 in front of the water molecule, and then you're set. So this is the reaction that's taking place in this problem. Now let's look at what we're given and see if we can solve. So we've got a volume of sulfuric acid, 13.84 milliliters. And we've got some more information that follows. 25 milliliters and 0.1 mole per liter of sodium hydroxide. And then it asks what's the concentration of sulfuric acid. So that means we need our concentration here. Now looking at this, once we've written down our balanced equation and look at what we're given, the problem-solving process should look very similar to types of stoichiometry problems we've done already. The only difference here is you're just dealing with acids and bases. Other than that, all the same kind of steps and procedures and ways to look at problems will be almost identical. So let's make sure we all know what's going on. So if we need concentration of our acid, remember concentration is moles over volume. So we need to find the moles of sulfuric acid, and then we're going to divide it by the volume to find the concentration. So we have to start with information from our sodium hydroxide, and then we'll be able to get to information with our sulfuric acid. So what am I going to do? First thing I need to do is find out how many moles of sodium hydroxide I have. We'll map this out up here so we can keep it as reference. Then I'm going to need to find the moles of sulfuric acid. And then I'm going to have to find the concentration of sulfuric acid. So let's walk through those steps one by one together. So first thing I want to do is find my moles of sodium hydroxide. So when I rearrange my formula, C is equal to N over V, I know I need concentration times volume. So that's going to be 0.1000 moles per liter. I multiply it by my volume. But remember, this is milliliters and these are liters. So make sure that you change this into liters. And then when you plug that in your calculator, you should get 0.0025 moles. So we have some moles of sodium hydroxide. Next, I need to convert them to moles of sulfuric acid. One substance to another, that's where your mole ratio is going to come in. So, how many moles of sulfuric acid do I have? So I need my mole ratio, and for my mole ratio, I need my balanced equation. So that's why we started off with it at the beginning. So there's my moles of sodium hydroxide. I've got 1 mole of sulfuric acid for 2 moles of sodium hydroxide. So essentially what I need to do is divide by 2. So when I punch that in my calculator, 0.00125 moles of H2SO4. Now that I've got my moles of my acid, now I can find the concentration. Remember back up here we were given the volume of our acid. Now I have the moles, so I'm going to combine those two things together. So, step 3, last one, find the concentration of sulfuric acid. So, C is equal to N over V. So I take my moles that I just calculated and I divide it by my volume, and make sure that you change this into liters. And then let's take a look. We get 0.090317 moles per liter. And then we're going to round to significant digits. And for this question, we need 4. So I need to keep 9, 0, 3, and then I'm going to round the 1 to 2. And that's how we solve some neutralization problems with acids and bases. So looking at the steps, you can probably see they're very, very similar once you get started to your solution stoichiometry problems. Also, you could combine all 3 steps together in a unit analysis method for solving the problem if you like. You don't have to break it apart into 3, but if you're happy with the 3 steps, then stick with that if you know what's going on. Before we try out some more problems, we're going to talk about something called titration. So what is titration? So let's talk about in general what it is first, and then we'll look at some equipment. So titration is an experimental technique. This is part of analytical chemistry. If you're interested in analyzing things, this is part of a branch of chemistry called analytical chemistry. And in general, what it's used to do is find the concentration of an unknown solution. You have a sample, you want to know what its concentration is. You've probably done some analysis on already to have an idea of what it is, but you need to find the concentration. It works really well with acids and bases, but it's not limited to them, but that's a very common application of it. So if you're talking about an acid-base titration, the reaction that's occurring is neutralization. An acid will react with a base, and you're studying that reaction and any measurements you get in that particular experiment. So what we're going to do is go over the general setup and some parts for titration and a little bit more theory behind it. And then we're going to try out some of the calculation problems. And then you'll have a chance to review some of the titration actual technique and videos and virtual lab opportunity another time. So let's take a look at this on more of a full view together. So this is the setup. Let's go over the equipment first. So this piece of equipment here you're probably familiar with. This is a retort stand. So there's your retort stand. It's going to hold all of the apparatus together. This guy up here is a clamp. This piece of equipment you should also be familiar with. That's an Erlenmeyer flask. This piece of equipment up here, it's very long. It's probably about 70 centimeters long, 80 centimeters long. It's made out of glass, and it's called a burette. So think of a graduated cylinder, but it's a lot more narrow. And if you check out some of the videos that are posted, you'll see some of the equipment in real time to get a better sense of what it looks like. So this is a burette, a more narrow type of graduated cylinder, but it's a little bit different. It's narrow. The markings on it are very small, and there's a valve down here that limits how much of the solution in it can go into the flask. So this valve, or you could say tap, there's a few names for that one, but essentially it controls how much goes out of the burette or shuts it off completely. Then there are some solutions that go inside of these pieces of equipment. So in the burette, it says here you have a titrant. We'll write this out a little bigger. So titrant goes inside. The titrant is a solution of known concentration. So whatever goes inside the burette, you know the concentration of it. You've measured it, or you're using a solution that's already been prepared. Then you have another solution down in the Erlenmeyer flask. So this solution in here is your analyte. The analyte is what you're analyzing. You don't know the concentration. So it's a solution of unknown concentration. You also need to add something to that solution. So in there we put an indicator. So one of those chemical indicators that you might have seen before, phenolphthalein is very common, or methyl orange. There's lots of them, but you need an indicator. So essentially what happens is you measure out a volume of your analyte, of your unknown, and you do that very precisely. You would do that with a graduated pipette. So if you remember making up solutions and seeing that pipette in the bulb, you would measure that out and put it in your Erlenmeyer flask. Then you would add a couple drops of your indicator. Why do you do that? Usually your two solutions, your acid and base, have no color to them. So the indicator is going to help signal when the reaction is done. And then what happens is you essentially let some of this solution come out at different rates while mixing the flask. And once your solution inside your flasks turns to a particular color, it signals that your neutralization reaction is done. You shut off the valve and you can take measurements along your burette of how much actually came out. So what's your starting point on your burette? What's your final point on your burette? And then you can take a change in volume to find out how much actually comes out of it. And then you'll have data to do some analyses. So we'll look at some data in a little bit of how that works. You can put the acid or base inside the burette or the flask. It depends on what you're analyzing. Often the base is the solution where you know the concentration, so it goes in the burette. And, and the unknown is often an acid, so it goes in the flask. But it's not limited to that. You could have it the other way around. But you can't have two acids and two bases. One of the solutions has to be a base, and one of them has to be an acid. So at this point, it might be a good idea to pause the video and go and take a look at one of the titration technique videos that are there, just so you can see in real time what titration is all about with the equipment, just so you can understand the next couple of questions a little bit better, or what some of the terms are being referred to, and then pop back and we'll look at a few more terms and try some problems. So down here at the bottom, something that we didn't just cover, is about titration. So we said in a titration we're going to have an acid and a base, and the goal is where we want the amount of moles of the acid to equal the moles of the base. So in our titration, we want moles of our titrant to equal to the moles of our unknown, our analyte. That particular point has a special name. It's called the equivalence point. Equal amounts of moles, equivalence point. And that's according to your balanced equation. We need to make sure that we signal when that equivalence point takes place, and this is where the indicator comes in. In a titration, the point when the indicator changes color is called the end point. When you're doing a titration, what you want to make sure is your equivalence point is as close as possible to that endpoint. So what does that mean? You want the number of moles that are going to react to be equal when the color change occurs. You wouldn't want your indicator to change color too early if your reaction isn't complete. How to choose a proper indicator takes a little bit more knowledge, which is something for a future chemistry course in grade 12. But for now, we just need to be familiar with those terms. We want the moles, equal number of moles to react with one another, and we want the color change for the indicator, that endpoint, to happen at the same time. So what we're going to do is try a couple of problems where they're using some terms with titration and some data. Once we break that down, hopefully you'll be able to see that the problems are very similar to the first example that we looked at when we were looking at acid-base neutralization. So let's try this one out. So here we've got 25 milliliters of an unknown solution of sodium hydroxide. Is titrated with 63.5 milliliters of 0.725 mole per liter hydrochloric acid. What's the concentration of sodium hydroxide? Here, the key word that's a little bit different is titrated. But if you hear titrated, remember titrated means acid base neutralization. So it was performed in the lab using the equipment that we just went over through a titration, but the actual chemistry that happened was an acid-base neutralization. So let's write out an equation for this reaction, and then we'll look at what's going on. We've got sodium hydroxide, then we've got hydrochloric acid. We're going to do our double displacement, water and salt, so H2O. And our salt here is made up of sodium and chloride ions. And this one's already balanced. It's 1-to-1 ratio, so it's nice and easy. So we have a volume for our sodium hydroxide, but it says it's the unknown. So in this case, the sodium hydroxide is your analyte. I'll just put this in bracket to practice some terms. We have a volume of 25 milliliters, but we don't know the concentration. Here, we have a solution of hydrochloric acid. We know the volume and concentration. So this is our titrant in this example. That means it's going in the burette. The volume that was used, so this was already measured from your burette, is 63.5 milliliters, and the concentration is 0.725 moles per liter. Now, take a step back and look at what you've written down for your given information. Even though it sounded a little bit different, look at what you actually have to do. You need to find the concentration of a base. You have information about the acid. Think back to that first example we went over. Now that we've written it down, it should look very familiar to you. It's the same types of steps and same stoichiometry approach, even though the data came from a slightly different place. So I want you to try this one out on your own just to test yourself to see if you know what's going on. So pause the video, and then we're going to walk through the steps together. All right, let's take a step back, look at what we've got, and then we'll look at the answers. So I start off with moles of I start off with volume and concentration of hydrochloric acid, which means I can find the moles. So the first thing I need to do here is find my moles of hydrochloric acid. Again, I'm going to use my concentration and volume equation. So I take my concentration, 0.725 moles per liter, and multiply it by my volume. Again, make sure you change to liters, and you should get 0.0460 moles. Next, we have to change our information about hydrochloric acid to sodium hydroxide. It's a 1-to-1 ratio. So if you want to take a shortcut when showing your work, that's totally fine if it's 1-to-1, but you need to show the ratio somehow. Since they're both reacting equally, I know my moles of hydrochloric acid reacting have to be the same, also 0.046 moles. Now I have my moles of acid, I have my volume of acid, I can find the concentration. So when I do that, I take my moles, divide it by my volume, and that's missing here, so let's make sure we add that in. And then we get 1.8415 moles per liter. And then make sure we round to significant digits. So for this one, we need 3 significant digits. So we get 0.184 moles per liter. You might have noticed in some of the problems that instead of writing moles per liter, there's another symbol that's used. You can do this as well. If you want to do this, you sometimes see a capital M being used. This is used for molarity. So it's implying moles per liter. So instead of always writing out moles per liter, you can write that in. But I would just do that at the end. I wouldn't do it in the middle of calculations because it's easy to mix up some of the units. All right, one last problem, and this is going to look at some actual titration data. So here we've got a titration carried out. It's using a 2 mole per liter standard solution of sodium hydroxide. And the titration was carried out with 10 milliliter samples of sulfuric acid of unknown concentration. The titration is performed 3 different times, and observation and data are recorded in the table below. And we want to find out what's the concentration of the sulfuric acid. So let's take a look at the data table together, and we'll see if we can break this down. So in the first column here, we see volume of sulfuric acid. There are 3 times this experiment was performed. Each time you're using the same volume of acid, 10 milliliters. The next 2 columns come from readings from the burette, that long cylindrical glass tube where you take your measurements. A starting and final reading of your base. Then over here we've got a column we can fill in for the volume of sodium hydroxide that was actually used, and then we'll need to find an average. But before we actually do that, let's make sure we understand the reaction. So the reaction is between sodium hydroxide and sulfuric acid. This is going to give us our water and our salt. And then make sure it's balanced. Okay, so even though this question sounds a lot different and has a lot going on, this is still the reaction that's taking place. A titration means acid-base neutralization. We've got our base and we've got our acid. In the question, if we take a look back up at the top, there's a key word here that was used. It said standard solution. Standard means we know the concentration of it, and we were told it here. So here, the sodium hydroxide, for reference, this is our titrant. This is what's going in the burette. We know the concentration of it was 2 moles per liter. And if you like better just writing moles per liter, you can also write that. Then we have sulfuric acid. In the question, it says this was unknown concentration, but every time the experiment was done, we have 10 milliliters for the volume. Since this is our unknown, this is our analyte. This is what would go in your flask. Every time we do this experiment, the same volume is used, so we can note that down here, 10 milliliters. We want to find out what the concentration of our acid is. This is the purpose of titration. What's the concentration of our analyte? But we need to get some data for some sodium hydroxide. So far from the question, we only have the concentration. So this is where the data table comes in. So let's move that back up and take a look at this for a moment. So in our equation below, we've taken into account how much acid was used. Experiment was performed 3 separate times, every time the same volume was used. When we look, though, at our burette readings, we've got two numbers, a starting point and a final point. Here, if we want to find the volume of sodium hydroxide used, we need to do the change in volume. Our final volume, which is here, minus our initial volume, which is here. Where did we finish on the burette? Where did we start? So change in volume here, we have to do 27 minus 10, makes it easy, 17 milliliters. So the first time the experiment was done, they needed 17 milliliters to neutralize that volume of acid. It was performed again. The final reading ended up being 31.5, but it started at 15. So 31.5 minus 15, we get 16.50 milliliters. Then we have to do the same thing for the third trial. We need that volume. Final minus initial. This one also ends up being 16.50. So we've got slightly different volume for the first, but two for the second and third that are the same. When you're doing an experiment, usually your data varies from trial to trial because of accuracy and some experimental errors that happen, and that's why you see different values being produced here. But if you're pretty happy that you've done the best job you can without making any human errors along the way, you keep your data, and all the data is pretty close. And to make it more accurate, this is where the average comes in. So when we see average down here, we want to average these three values together. So 17 plus 16.5 plus 16.5, 3 trials, we divide it by 3. So the average of these 3 is going to be 16.67 milliliters. So that means the average volume of base of sodium hydroxide that was used to neutralize 10 milliliters of our acid was 16.67 milliliters. So now we have some data we can use to help us solve our problem. So down here, we need a volume for our sodium hydroxide, and that's where the average number is going to come in, 16.67 milliliters. So let's make sure everyone remembers where that comes from. This is our volume here from the average. We're taking it from our experimental data. So now that we've taken our data from our titration trials, now we can take a look at our balanced equation and the data that we have. We're going to completely ignore the rest, which is why I've moved it out of view here. Now take a look at the scenario we have. I have information about my base, I need to find information out about my acid. Does this look familiar again to the first two problems we've already seen? I hope it does, because once you've sorted out your data like we did, the math and problem solving is the same. So, what are the steps I need to do? Similar to before, I need my moles of sodium hydroxide. Once I have that, I need my moles of acid. And then I need to find my concentration of acid. So, using the data, we can find moles of base. We've got our mole ratio to find the acid, and then concentration. So why don't you pause and see if you can solve this problem. Find the concentration of sulfuric acid. And then when you're ready, we'll walk through the answers step by step together. All right, let's get solving. So first thing I want to do is find my moles of sodium hydroxide. So concentration times volume. I've got 2 moles per liter. And I'm going to multiply it by my volume in liters, so 0.01667 liters. Let's move this up a bit. So 2 times my volume, I get 0.0033334 moles. Next, I need to do my mole ratio step to find out my moles of sulfuric acid. Now let's go back up and look at our balanced equation here. It's 2 moles to 1 mole. They're not equal. So I have to include my mole ratio and my units. Same 3 in there. Okay, and then I've got 1 mole of acid for 2 moles of base. So in my calculator, 0.03334 divided by 2, I'm going to get 0.01667 moles of acid. And then I need to find the concentration. So C is equal to N over V. This is for sulfuric acid, 0.001667 moles. And I'm going to divide it by my volume, 0.01 liters. That's that 10 milliliters. And then I get 1.667 moles per liter. Then I want to make sure that I round it to the correct number of significant digits. So going back and looking at my question, I have 2.00 for my concentration. So I need to round this to 3. So 1.67 moles per liter. And that's the concentration of sulfuric acid. There are some questions that might get you to do even a step further. They could ask you, once you have the concentration of your acid, what's the pH? So if you needed to do that, then you would need to do an extra step where you're finding pH is equal to negative log concentration of your hydrogen ions, and you would need to find the hydrogen ion concentration in here. So that's something that you could do as well. That would be an extension on this type of question. So after today's lesson, the big focus is practicing the stoichiometry and calculations and getting a general idea of titration And then the next part will be learning more about titration, getting familiar with the setup, getting familiar with the technique, knowing the names of the equipment and the purpose of it in more detail, and so on.